on the screen. If you could scan that, if you've got a smartphone, and complete the pre-webinar survey, we would be really grateful. It'll only take a couple of minutes while I'm just going through the housekeeping. So yeah, we are recording this session so that people who aren't able to attend will be able to watch it afterwards. So we're going to upload the recording onto our YouTube channel and our website, and we will send a link to the recording to anyone who registered for the webinar today. And we would encourage you to share it with your colleagues as well. It will be available to everyone. So because we're recording it, please can you turn off your camera, ensure that your microphone is muted during the presentations. Otherwise, any conversations that you're having in the background might appear on our YouTube video, and you wouldn't want that. So yeah, camera off, microphone off. And if you've got any questions, please put them in the chat. There will be an opportunity to ask questions at the end. If we don't get enough time, then we will work out a way to respond to those questions and either email out or put something on our website afterwards. So I'm going to start the webinar officially. So I know there's a few people just joining us. So just a reminder, we are recording today. So welcome everybody to our webinar, and the spotlight today is on common aneuploidy testing. My name is Karen Creed. I'm lead midwife for genomics in Central and South Genomic Medicine Service Alliance, and I'm going to take you through what the next hour looks like. So we have got quite a packed hour for you. I'm going to start off with R445, and we are assuming that most people joining the call today will have a fairly good working knowledge of R445 and of non-invasive prenatal testing. But just in case you're not really sure what we're talking about, I am going to give a very brief overview as to what R445 is and what NIPT is. And then I'm going to hand over to Nikki Jones. Nikki is one of our genomics trainees in the labs at Birmingham Women's, who is going to present an audit of the data from the first year of implementation of R445, so we can see how it works in practice. And what we hope is that some of this data will help support the conversations that you're having with women and their families in your care. Then, just to give it some clinical context, we're going to hand over to Marianne Quinn, who is lead midwife for genomics in the South East, and also NIPT lead midwife at the Safe Lab at St George's in London. And she's just going to present a case study and also just give us some of the feedback that they've received from service users and from health professionals since R445 has been launched. Then we're going to change tack and we're going to look at QF PCR. We've got Beth Young, who's our principal clinical scientist at Birmingham Women's, who's going to give an explainer on QF PCR, just a reminder of what it is, but also discuss some of the challenges that we may encounter with confined placental mosaicism and how we interpret those results. And we know we're getting a lot of questions about that. Following that, we'll signpost you to some resources and further information, and then Q&A at the end. So a lot to get through, and I'm going to move straight away and start looking at R445. So what is R445? Well, that is the clinical indicator code in the National Genomic Test Directory. And that test directory, of course, outlines the full range of genomic tests that are commissioned for the NHS in England. So for each indicator code, the directory also gives details of who is eligible for that test, what the testing methodology is, and which clinical professional can request that test. Now, the directory is regularly updated. It's available online, and you'll see that I've just I put the link down at the bottom, but if you Google genomic test directory, it will come up. And what we recommend is, because the directory is regularly updated, which I think reflects the fact that genomics is changing rapidly, then we always recommend that you check the directory before ordering any test, just in case there's been any changes, particularly to eligibility and exclusion criteria. So R445 specifically offers NIPT, or non-invasive prenatal testing, to any woman with a history of a pregnancy with full trisomy 21, 18 or 13. So remember, it's not available for women who have a previous translocation or mosaicism. And the rationale for offering R445 to this group of women is that they're known to have an increased chance of any primary trisomy in a future pregnancy. So with R445, we are offering an opportunity to proceed direct to NIPT, which is a more sensitive test and performed at an earlier stage in pregnancy than the current first-line screening tests that are available on the fetal anomaly screening programme. Now, R445 was implemented across England in April 2024, and I know that some of you may have been offering it to this group of women prior to that, but it was only universally offered across the country from April 24, so that's when we're going to look at the data from. This next slide, I think, will probably be familiar to many people on the call. So this is just a reminder of what NIPT is. And of course, in NIPT, what we're looking at is cell-free DNA, or cfDNA. And this is fragments of DNA that's released into the bloodstream after a cell has died. Now, during pregnancy, of course, we have cfDNA from both the mother, but also cfDNA that originates from the placenta. In most cases, this placental cfDNA is going to reflect the DNA of the fetus. Now, it's usually detectable from the placenta from 7 weeks of pregnancy, but of course for NIPT we need to make sure there's enough cfDNA from the placenta, and therefore we have to wait until 10 weeks, so we've got enough that we can analyse. And what we're measuring is the relative quantities of cfDNA fragments in the maternal blood. And where we see a higher relative amount from a specific chromosome, then that could indicate trisomy. But remember, NIPT is a screening test, it's not diagnostic. And the presence of trisomy can only be confirmed by prenatal diagnosis. So this is just a reminder of, or sort of an overview really, of the R445 pathway. So think of R445 as a pathway rather than just a test. And anyone who meets the eligibility criteria for R445 should be offered the following 3 options. They can choose, obviously it's their choice, so they can choose to not have any screening or diagnostic tests. They can choose to have NIPT on the R445 pathway, or they can opt direct for prenatal diagnosis if they want to know for sure, but bearing in mind, of course, that that carries a risk of miscarriage. So where women or people do accept R445, then it's essential that they have a dating scan, because we need to ensure that the blood test is taken at the correct gestation, and that's any time between 10 weeks up until 21 plus 6. Of course, just a reminder, if there are any anomalies at that scan, then refer to fetal medicine. Don't take R445 because it might not be the most appropriate test to take at that point. So results for R445 NIPT, then they're reported in the same way as results, NIPT results on the fetal anomaly screening pathway. So if they get a low chance result, then pathway stops at that point and they go back to routine antenatal care and are offered a 20-week scan, of course. They may get a no result. So if we get a no result, then they can have 1 repeat NIPT or prenatal diagnosis or no further tests. And of course, if that second NIPT sample also fails, then at that point they don't get offered another NIPT test, but they can go for prenatal diagnosis if they request, or can opt out at that point. Or of course, they may get a higher chance result, in which case
refer off. I'll highlight it.
Oh, can everyone just go on mute, please? Thank you. So higher chance result, then they get the offer of prenatal diagnosis. So remember, the offer of R445 is replacing the offer of combined and quadruple screening on the fetal anomaly screening programme. Remember that combined and quadruple tests have lower sensitivity than R445. So we're offering this group of women a more sensitive test and at an earlier stage in pregnancy. So I'm now going to hand over to Nikki, who's going to talk more about the data from the first year of testing. So Nikki, are you there? Hi Nikki, you're on mute.
Sorry. Now. So yeah, I did an audit on the R445 non-invasive prenatal testing pathway for the first year of the service going live. And the names below on this slide are just all the people that helped with the audit, whether that was gathering data or giving me advice of how to complete it. Next slide, please, Karen. So I don't want to repeat what Karen has said, but essentially NIPT is for Downs, Edwards and Patel syndrome, is now available as part of the fetal anomaly screening pathway for those women who have had a previous pregnancy with a reported full trisomy of chromosomes 13, 18 or 21 under the NHS test directory code R445. So I performed an audit on the R445 NIPT service, which included data from Birmingham Women's Laboratory, the TDL Genetics and Health Service laboratories and St George's Hospital to determine how many patients were referred in the first 12 months of the service going live and what the outcomes of this testing was. Next slide, please. So the questions that we essentially wanted to answer from this audit included how many women accessed this service between April 2024 and March 2025. How many of these women then went on to receive a high chance NIPT result and for which trisomy? What was the trisomy of their previously affected pregnancy? And then did these women go on to have invasive testing and what was the outcome of this? And what was the average maternal age and gestation of those women referred at sampling? Next slide, please. So now we will just be moving on to me showing you the data from the first year post-implementation. So between April 2024 and March 2025, we received a total of 1,840 referrals for R445 were received nationally, with similar referral patterns seen at all three laboratories. So this is demonstrated in the pie chart on the slide. So as you can see, TDL Genetics received the most referrals at 681, closely followed by the West Midlands Genomics Laboratory, which received 633 referrals, and then followed lastly by St George's, who received 526 referrals in total. All results were obtained using either the Illumina VeriSeq NIP Solution 2 or the Iona prenatal test. Next slide, please. So among the women that were referred through the R445 pathway, 1.8% of these women went on to receive a high chance NIPT result, with 48% of these cases involving the same trisomy as in their previous pregnancy. In cases where we did have access to the invasive test results, either CVS or an amniocentesis, the NIPT high-risk findings were confirmed in 85% of these cases. So in this table below, you can see that in total, we received out of the 1,840 referrals, 33 of these women received a high chance NIPT result. So 22 received a high chance trisomy 21, 6 received a high chance trisomy 18, and 3 received a high chance trisomy 13. So in total, that's 1.8% of total referrals. Where we did have access to follow-up test results, you can see that the women that received a high chance trisomy 21 and a high chance trisomy 18, the invasive test confirmed this in 100% of cases. Whereas, as you can see, with those women that had a high chance trisomy 13 result, the invasive had a disconcordant result with the high chance NIPT result. Next slide, please, Karen. So then we just looked at what the average maternal age of the people that were accessing this service were. So as you can see across the whole cohort, the average maternal age was just under 35 years. And those who received a high chance NIPT result had an average maternal age of just under 37 years. And then this bar chart just demonstrates the average maternal age separated into the 3 trisomies. So you can see that the average maternal age was higher in those women that had a high chance trisomy 21 result, and the average maternal age was the lowest in those women that had a high chance trisomy 13 result. Next slide, please, Karen. And then we just wanted to look at what the average gestational was at referral. So this was across the cohort was 12 weeks. And then the mean gestational age at referral in those women that received a high chance result was 11.2 weeks. And then this bar chart again on the slide just shows that the average gestational age at referral was highest in those women that received a high chance trisomy 18 result. And the average gestational age was the lowest in those women who received a high chance trisomy 13 result. Next slide, please, Karen. So then just to summarise what this audit essentially has told us, so between April 2024 and March 2025, so a year post-implementation, we received a total of 1,840 referrals for R445 nationally. 1% of these women went on to then receive a high chance NIPT result. Although a small number of these were unable to be confirmed invasively, this figure is in line with the published literature that having a trisomic pregnancy increases the risk to future pregnancies to 1% above the age-related risk. So it does confirm what the published literature tells us. Where available, the invasive testing confirmed 85% of the high chance NIPT results. Maternal age across the cohort on average was just under 35 years and just under 37 years among the high chance NIPT cases. And then the mean gestational age across the cohort at referral was 12 weeks. And then this was decreased to 11.2 weeks for those that went on to receive high chance NIPT results. Thank you. That's the end of my slides.
Thank you, Nikki. That's a phenomenal amount of work that you've done there, because over 1,800 cases to review. So thank you for presenting that. I mean, I think it's really important that we have this data, especially, around the sort of like chance of getting a high chance NIPT result for this group of women, and that, this test is really sort of bringing benefit. But I think what we'll do is Beth, you've got your hand up. Sorry, I can't see everyone's hands.
No, sorry. I know we want to move on, but it was just a comment that something I thought was quite interesting in Nikki's data, of the ones that weren't confirmed, the 3 cases that weren't confirmed, they were all trisomy 30, were they 30? Yeah, and what we do find, so sorry, I'll introduce myself properly in a minute. I oversee the NIPT service here in Birmingham. What we do tend to see is a higher percentage of false positives in trisomy 13 and 18, and that's partly due to the test, but more often due to confined placental mosaicism. So that will fit in really nicely with what I'm going to show you in the final part of the presentation. So it was just an observation from them and an IPT person.
No, that's great. It's really interesting to point that out. Thank you. I think what we'll do is we'll go into Marianne's presentation on her case study. Are you there, Marianne?
Yeah.
Lovely. So if I hold on. Sorry, just find my cursor. There we go. So I'll hand over to you.
Thanks, Karen. Thank you. So my name is Marianne, and as Karen said at the beginning, I am the lead genomic midwife for the South East, and I'm also the NIPT midwife at St George's Safe Laboratory. So where Karen and Nikki have very helpfully sort of shared some of the background to the pathway and the data that's been collected over the last year, thought it would be helpful just to share a case study from practice. So as part of my NIPT role, we follow up all the outcomes for our NIPT testing, and we also gather some feedback from the women and the midwives using the service. So again, just highlighting this in a real life scenario, how this pathway has been implemented and the difference that it's making. Next slide, please, Karen. So the name has been changed, but for the purpose of this, we've got Marie. And this was her second pregnancy, and in her previous pregnancy, she received a higher chance combined screen test result for Down syndrome of 1 in 44. That's just over 13 weeks. She, as part of the fetal anomaly screening programme, she opted for NIPT, and she received a higher chance NIPT result for T21 at just over 14 weeks. She chose to proceed with an amniocentesis for diagnoses, and that was carried out at 15+4, which confirmed that baby did have trisomy 21 or Down syndrome. And Marie made the very difficult decision to terminate this pregnancy at just over 16 weeks pregnancy. So she is now pregnant again, and she books in with her midwife at 9 weeks, and the community midwife is aware of the R445 pathway, so she refers into the screening team for further discussion. And just after that appointment, she has a telephone appointment with her screening midwife who talks through the pathway, the options available, and Marie chooses to proceed with NIPT as a first-line screening test as per the R445 pathway. So at 10+1 week, she comes in for a early dating scan, and those bloods are taken and sent, and she consents for screening for trisomy 21, 18 and 13. And a few days later, she receives a subsequent high chance NIPT result for T18. So sort of this is one of the things that Nikki found in her audit, whereas Marie had a pregnancy affected with Down syndrome in her previous pregnancy. She's received a higher chance result for Edwards syndrome in this pregnancy. So following this NIPT result, she is seen in fetal medicine. She has a detailed scan, and there are some anomalies seen on scan. She opts for a CVS. The CVS is taken at 11+3, and this confirms T18 in the fetus, which is in line with the NIPT result and the scan findings. So Marie's had a very difficult obstetric history, and I suppose that she's gone through a similar experience in this pregnancy as she did in the previous pregnancy. But a big sort of difference here is she was 11+3 when she got this information in this pregnancy. In the previous pregnancy, she was over 16 weeks. So that can be a sort of a huge emotional difference for these women. May seem like a short period of time for me at the moment anyway, but 5 weeks is a big period of time for women when they're going through these screening pathways. So this was just to sort of highlight, firstly, how women are experiencing this pathway. You're able to get results earlier, and also again that as I said, that Nikki had said that a previous trisomy 21 pregnancy does not just increase the chance of having a trisomy 21 pregnancy, but that it increases the chance for all 3 common aneuploidies. So if we can go on to the next slide, please, Karen. And just finally, I was going to share some of the feedback. So as I said, we gather feedback from the screening midwives and from the families who use NIPT through us at St George's. And it's been really wonderful reading this feedback, and I'm just sharing some of the snippets with you today. So from the screening midwives perspective, they said our families are absolutely delighted to be offered this test. And again, really happy with the introduction of R445, excellent service to women and their families who've experienced a previous pregnancy. And then more importantly, that feedback from the families who have been using the R445 pathway. So one family said, my anxiety was through the roof. I know this is not a diagnosis, but feel so much more reassured and relaxed now. And another, I am so grateful for the screening. With a previous pregnancy with a diagnosis of Down syndrome, the assurance of the test was crucial to my mental well-being. And then finally, this time the chances were low, but that was not the case last time. The information was very clear and the team were tactful and professional about what the options were, explaining in detail, but in a gentle and compassionate way. And I thought I'd finish on that one because that's actually what I see over and over and over again with our feedback surveys, the care, compassion and information that you as midwives are providing these women is just outstanding, and it's really nice to sort of hear that. But that's just a little bit of a case study and feedback, so hopefully that's been useful.
Thank you, Marianne. That's amazing. And I think, the fact that it's now available across the whole country now to all women who meet the eligibility criteria is a huge step forward and clearly bringing benefit to a lot of people. Now, I can't see the chat, so I'm not sure if there's any questions about this specifically, or we can wait to the end. If there's anything easy to answer now, Marianne, is there anything in there? If not, we'll go on to Beth and then we'll do all the questions at the end.
There's a reasonably, I can answer Michelle's question. You should have, depending on where your NIPT referrals go, you should be given the sensitivity and specificity information on the report for the NIPT. So I can comment on what we do here in Birmingham, but I don't know where you send yours, but there will be a specificity and sensitivity comment on the report. We don't yet have the information for how NIPT has been used as part of the fetal anomaly screening pathway. So the sensitivity and specificity information we share is that on the general population. We are still waiting from the FASP team the information on how NIPT how well it works in women who already have an increased chance of a trisomy. And we're hoping that will be soon, but it's a lot of statistics to get through.
Thank you. Should we move on to QF PCR? Beth, are you going to share your slides?
I will try.
Let me stop sharing.
I'm hoping that's now worked.
Something's coming up. Yes, I can see it. That's lovely. Thank you.
Hopefully it'll move on in a moment. So yes, I'm Beth Young. I'm the Principal Clinical Scientist in the Prenatal and Reproductive Medicine team here in the West Midlands Genomics Laboratory, and I oversee the day-to-day running of the NIPT service for our third of England. But I also have an interest in QF PCR and oversee that service as well. So QF PCR is really what will be performed as a follow-up to a high chance NIPT, and I want to go into that in a bit more detail because I know there have been a lot of questions, especially around some reports that have been in the media this year around false positive QF PCR results or false positive trisomic results following CVS. So has that moved on okay?
Yes.
Brilliant.
Thank
you. So what is QF PCR? It's simply quantitative fluorescent PCR, and PCR is polymerase chain reaction. It can be used in lots of different techniques, but where I use it in my job in the prenatal and reproductive medicine service is using it to look at prenatal samples such as amnios, CVSs, fetal blood or cord blood to look for common aneuploidies, i.e. trisomy 13, 18, 21 and monosomy X. Oh, don't know why am I. So I know you'll all be familiar with what an amnio looks like because it looks the same when it comes to us as when you take it. But I just thought I'd also include these pictures of what a CVS looks like once we've cleaned it up. So when we receive it, there's a lot of blood and in there. We wash it, clean it up, and we separate out the maternal tissue from the fetal tissue, the placental tissue. So this image here after cleaning is just pictures of the fetal, the placental villi. And that's what we will test in the QF PCR. Just to give a bit of background to QF PCR, historically, we performed a full karyotype on any prenatal samples we received to look for the trisomies. And a karyotype still is considered the gold standard because it looks at multiple differences in the genome, and they'll look at multiple cells. However, it's quite labour intensive and it can take up to 2 weeks to report a result. And so the introduction of QF PCR, which is a test that can be done rapidly on a very small sample size, allowed us to generate much quicker results for women. We usually report the result within 2 days. QF PCR has been being offered since 2003. Within 2005, the service extended to all CVS samples. So for 20 years now, QF PCR has been the frontline test for trisomy 13, 18, 21 for anybody who has an invasive test. And this rapid positive result allows better pregnancy management, whereas a rapid normal result gives faster patient reassurance. So really, the benefit of it is a really fast turnaround, 3 days, as Marianne was touching on earlier, the wait when you're in this pathway, the difference of 10 days to get a result is quite significant. So how does it work? So it's a PCR amplification assay that looks at highly polymorphic repeats on the chromosome. So these repeats are called short tandem repeats. This is just normal variation in the population within all of us. We identify these areas in the genome where you have a repeat sequence, usually 4 nucleotides that's just repeated over and over and over. And we all have a different number of repeats. It can range from 5 to 50. When you create the PCR, when you perform the PCR, the size of the PCR result, the PCR product you get will be directly related to the number of the repeats. I'll show you that on a slide a bit more in a minute. But say on one of my chromosome 21s for marker, it's really sad I know the names of the markers, D21S226, you don't need to know that. But one marker on chromosome 21, I have 50 repeats. On the other one of my chromosome 21s, I have 25 repeats. I'm going to get 2 different PCR size products representing that repeat. The way we do the PCR is quantitative. That's why it's called QF PCR. So the amount of product we get is directly relative to the amount of starting product. So we can say how much of each chromosome the fetus has. We have all these PCR products that we put together. We separate them through a capillary by adding an electrical charge, and they're all separated by size. And we add fluorescent tags so we can detect them. And I'll show you why that's important now. But before I do so, sorry, just to briefly touch on the kits we use. We have a multiplex kit that we use to look for trisomy 13, 18, and 21. And that's shown by the green the blue letters here on these schematics. And that consists of 5 PCR markers for chromosome 13, 5 for chromosome 18, and 5 for 21. These 15 PCR products are all multiplexed into one PCR to give us information across the whole of chromosome 13, 18, and 21. We also include a marker, the amelogenin marker, and this is just to say this is a marker on chromosomes X and Y that we use as a QC metrics to make sure that our fetus is the expected sex. We do also have backup kits. We have another 4 markers on each of the chromosomes, which we can use in unusual circumstances. And again, I'll show you why they're useful in a moment. So this is the test we would run for any pregnancy that came into referred to our centre. We would use 5 markers for chromosome 13, 5 markers for chromosome 18, and 5 markers for chromosome 21 in a single reaction to give us information for each of those markers all the way across each of the 3 chromosomes. We do also have a kit where we suspect that the fetus may have Turner syndrome, monosomy X, and this will look at the markers on chromosomes X and Y. And this will just count the sex chromosomes and tell us whether the fetus is XX, XY, or XO for Turner syndrome. So I said that those 15 PCRs are multiplexed together, run down a capillary and separated by size and their fluorescence detected. This is what the raw data looks like when it comes off the analyzer. It's just a mess of fluorescence. So across the bottom axis is the size. You can see lots of different peaks being separated at lots of different size. And up the side is the amount of fluorescence being given off. Obviously, you can't read anything from that. So we need a special kind of software called Gene Mapper that we use here in Birmingham, and it separates out based on colour and on size. So each of these grey boxes at the top represents a marker. The software can separate the peaks into the marker, and we can analyse to see how many copies of each of those markers the fetus has. So to get to how we analyse it, as I said, we all have variation in these regions that we're targeting with QF PCR. So say this is the D21S226 marker I'm looking at for me. On one of my chromosomes, I have a repeat size of 100. On one of my other chromosomes, I have a repeat size of 110. So when those PCR products are run on the capillary, you're going to get a PCR product of 100 base pairs and a PCR product of 110 base pairs, representing my 2 chromosomes 21. And because I have equal amounts of both chromosomes, they'll be in a 1-to-1 ratio in their peak height. So we can say I have 2 copies of chromosome 21 at this marker. Now we do try and pick markers that are as informative as possible within the population, but it's not guaranteed. Sometimes, say on another one of my markers, D21S145, on both chromosomes, the repeat size is just 100. Now I do have 2 copies of chromosome 21, but I can't differentiate between them. And so in this condition, you'd only get a single peak. And that's why we use 5 markers for each chromosome, because no matter how polymorphic the markers we're using are, you are going to get some of these markers that we call uninformative. You can't tell how many copies of the chromosome are present. And in order to report a pregnancy as having no evidence of a trisomy, we need to have at least 2 out of those 5 markers we've used informative in a 1-to-1 ratio. This is especially a problem in couples who are in consanguineous relationships, because they will quite often have shared ancestral haplotypes. So their 2 chromosomes might be have areas in common. And this is when we'll use those backup markers. I showed you for each chromosome, we have 4 backup markers. So we can test another 4 markers, making it 9 markers in total we'll test for a chromosome. More often than not, that will give us our 2 informative markers we need to say there is no evidence of a trisomy. If we don't, we will report that chromosome as uninformative. So that's all uninformative means. It doesn't mean there's something wrong. It just means the markers can give us the information we need. And so we will then go on to recommend a karyotype, because that is the gold standard test. But what we're looking for are these trisomic markers. So that seems to be animated. That's where the fetus has 3 copies of a chromosome. So it may be that if mum was, say these first 2 chromosomes are inherited from mum and mum was informative, fetus has a marker that's 100 base pairs, a marker that's 110, and dad has a marker that's 120. And in this situation, you'll get 3 peaks in a 1-to-1 ratio, because you've got 3 chromosomes equal numbers of each of those chromosomes, each with a different size repeat. And this is what we call a triallelic marker. So there are 3 different size repeats. I showed you, though, that mum could be perfectly healthy and 2 copies of her chromosome, but they could be the same. So she could have, fetus could inherit both of her chromosomes that have a repeat size of 100, a dad's 120. It's still representing a trisomy in that fetus, but you're going to get a 2-to-1 ratio. I hope that makes sense. So these are all trisomic results, but you either have a 1-to-1-to-1 or a 2-to-1. And I'll explain in a couple of slides why that's so important. So this is what a normal multiplex result would look for us once we put it through the genetic analyzer. You can see that for, I did count these earlier, how many have I got? 9 out of the markers, we have 2 peaks in an equal ratio, equal peak height. For one marker, this one here, D21S1435, it's uninformative, so we can't say anything about that. But we have 2 other markers for chromosome 21 that are normal, this one here and this one here. I'm hoping you can see my arrow. The first 2, D21S1437 and D21S11. So we can say for this pregnancy, we have at least 2 markers for every chromosome that are in a biallelic 1-to-1 ratio. So there is no evidence of trisomy 13, 18 or 21. If I go on to a result where it's a trisomic fetus, I've highlighted the chromosome 21 markers in red. You can see for all 5 of them, there's either 3 alleles in a 1-to-1 ratio or 2 alleles in a 2-to-1 ratio. The markers that I haven't highlighted for chromosomes 13 and 18 are all in a 1-to-1 ratio. So we can say confidently that this fetus has trisomy 21, no evidence of trisomy 13 or 18. But what I wanted to finish off with, now that I've given you that background, is something that we've recently had a lot of queries about. I know there's been quite a lot of coverage in the media earlier this year about some false positive results that have arisen due to mosaicism. So I wanted to give you some reassurance as to why our testing has moved on and how we caveat that in our report, so you know exactly what you're getting. So the reason why you might get a false positive result is a phenomenon called mosaicism. Now, in genetics, mosaicism is the presence of 2 or more populations of cells with different genotypes within an individual. Mosaicism can occur in any tissue, and particularly in the CVS, because this is a rapidly turning over tissue where lots of cell divisions happen quickly. The the DNA repair facilities don't always work as well because it's just such rapid turnover. We know mosaicism can occur in CVS in about 1 to 2% of diagnoses. And there are 2 scenarios that this can be. It can be a true mosaic in which the non-disjunction of the chromosomes happen quite early in embryogenesis. So mosaicism is present in both the fetus and in the placenta. So that would mean baby would be mosaic for a condition. But the one that really concerns us is confined placental mosaicism. That's where the trisomy has arisen after embryogenesis. So it's arisen just in the cells that are going to make the placenta, and it's confined to just that tissue. But if we're testing the placenta of a marker of the fetus, we're not going to get a genotype that's representative of the fetus. And part of the reason this is a challenge is because of the way we process a CVS. So when we take when this biopsy is taken, the tissue is taken from outside the placenta, where you have the placental villi in close contact with maternal tissue. And so any CVS we receive in the lab, we clean and sort before we use it. And that was the image I showed you earlier with that beautiful cleaned villi at the end. But this sample does still contain both outer trophoblast cells and inner mesenchymal core of the placenta, each of which are derived at different stages of cell division in the development of the placenta. Historically, labs used to just test a villi from this trophoblast area, and quite often they use a technology called FISH, which isn't looking at those ratios of the markers, it's just saying how many copies of a chromosome are present. We've known about confined placental mosaicism since the 80s, and it's really well documented. And it's known to be present in around 1% of all CVS samples. Like I said, that's quite high for a tissue, and it's because of those rapid turnover of the cells. But for the common primary trisomies, the ones we're interested in prenatal testing, 13, 18, and 21, it's only about 0.26% of CVSs will have mosaicism of the trisomy. In most of the cases that have been tested historically, where the QF PCR was false because it was a CPM is because testing of a really small region of the CVS has been carried out, and the majority of the material they were testing was these cytotroph blasts that I showed you on the previous slide. So if you imagine you've got this mass of cells in the placenta, and if you're only testing a very small part of it, you might just by misfortune pick up the area of the placenta that has acquired a trisomy. And interestingly, when the testing's been done by QF PCR, we've shown that these false positive, these CPM false positives, the cell line exhibited a 2-to-1 ratio, not the 1-to-1 ratio. And I'll show you why that's important in a minute. This has really helped us to understand how we need to process CVSs. And so now FISH is not routinely performed as a test, it's QF PCR, so we can tell whether it's 2-to-1 or 1-to-1-to-1 ratio. And instead of taking those single villi and testing just a small area of the placenta, we do a digest of the whole sample. So we're getting that mixture of the trophoblast and core, and you're getting a representation of the whole placenta. So it's a much more informative sample. So why is it important to have a 2-to-1 or a 1-to-1-to-1 ratio? This is because of where trisomy might occur. So there are two types of cell division. There's meiosis and there's mitosis. Mitosis is the cell division that happens in the majority of cells in the body. And you start off with 2 copies of the chromosome and you end up with 2 copies of the chromosome. It's an exact, it's like a photocopy of the cell. What we're interested in here is meiosis, which is the production of sperm and egg. And in those, you create gametes in which the chromosome number is halved. Now, column A here is meant to show normal meiotic division. It goes through 2 divisions. You started off with a cell with 2 copies of each chromosome, and you end up with gametes with 1 copy of each chromosome. If an error occurs in meiosis 1, you'll end up with some gametes that have no chromosomes and some gametes that have 2 copies of each chromosome. Or if the error occurs in meiosis 2, you'll have some gametes with no chromosomes, some with 2 and some with 1, as you'd expect. The problem with this is if you've got those gametes that have been caused through an error in meiosis 1, the 2 chromosomes are going to be different. So yeah, sorry, I'm trying to think. This is really complex and I'm trying to make sure I explain it properly. If the error has occurred in meiosis 1, you're going to end up with 3 different chromosomes in total in your zygote. 2 from mum and 1 from dad. And each one of those should be different. And that's how you get that triallelic marker. If the error has occurred in mitosis 2, the 2 chromosomes in the egg will be identical, joined by a 3rd one from dad, and so you're going to get the 2-to-1 ratio. I have to try and get my head around this every time I go through it, so please don't worry if that doesn't make sense to you. What I want you to take away from this presentation is you can only get triallelic trisomy if the error has occurred in meiosis, so the cell divisions that form egg and sperm. So if you've got a triallelic trisomy in your fetus, you know it's happened right at the beginning in the egg and sperm production, so it'll be present in the fetus. Biallelic trisomy can arise during meiosis, as shown here, or mitosis, normal cell division. So you don't know if it's occurred during the production of the egg and sperm, or if it's occurred later in the division of the cells going into the placenta. And this figure I find really useful because it shows us how many cell divisions happen between the original zygote conceptus here at 1 through to at the top here, the undifferentiated cells dividing and dividing and dividing to form your CVS preparation. But if you compare that with the actual fetal sample down here at the bottom of the chart, there's been a huge amount of mitoses between those different cell types. And that's why you can acquire additional trisomies. So the takeaway I want you to get from this is if we have a QF PCR result where all the markers show a trisomy, but that only in a 2-to-1, there is a small possibility that that's due to the trisomy being restricted to the placenta. What we will do, again, in this situation, we would always try those backup markers as well. So it's very unusual for us to get a 2-to-1 trisomy only. If that does happen, though, we will flag it on the report to be extra cautious, to say that although we've detected a trisomy, it might have arisen postzygotically. It might have arisen just in the placenta. So you need to consider that. And we would recommend that unless the pregnancy has scan findings consistent with a trisomy, you wait for the karyotype result, which is going to be the gold standard result, before taking any further actions. We've recently here in Birmingham changed all our reports. So we used to only say if it was biallelic. We've added it in now for our standard report to say triallelic trisomy, to really try and add that confidence in the result. So as I said, the one message I want you to take away from this, the reason we talk about triallelic and biallelic is if it's triallelic, it must have arisen really early on in embryogenesis. If we've only got biallelic markers, we can't exclude the fact that it's arisen in the placenta itself and isn't present in the fetus. And obviously we appreciate that very, very big decisions are being made on these results. So we want to make sure we have them as accurate as possible. So in conclusion, QF PCR is a quick and reliable test to use to look for aneuploidy of chromosomes 13, 18, 21 and monosomy X. It is in most instances a standalone diagnostic test, but if we do have any concerns that there's with CVS samples that what we're looking at is representing the placenta but not the fetus, we will recommend waiting for a karyotype. And there are complications that need to be considered, so sometimes the result won't always be straightforward. But if you're not sure, if you're not clear about what the result means, I would always urge you to contact the lab. We're happy to talk to you and talk through what a result means. And we do have best practice guidelines that exist to assist us in reporting the more complex cases. So all the labs will be referring to these best practice guidelines and we'll all be reporting in the same way. I hope that made sense. I know it was really complex. I've been working in this field for 15 years and it still makes my head hurt a little bit. So if you just take that one message away from that, I'll be happy. But please do let me know if there are any questions.
Thank you, Beth. It is complicated stuff, isn't it? We were talking about this earlier, and sometimes you just have to listen to it several times just to kind of get it into your head. But I think the message really is if it's triallelic, we can be confident it's in the baby. Yeah. If it's biallelic, there may be some doubt, and that's the take-home message, I think, isn't it? Yeah.
And we try and use as much information as possible. We have those 9 markers for a chromosome, so it really increases our chance of we only need one triallelic to be happy with it. But as I said, it's sometimes in couples, just by chance, you might not have a marker that's triallelic, but we just have to be cautious.
Thank you. We have got someone with their hand up. Hold on 1 minute. Sorry, I can't see your name. Is it Yatundi?
Yes, thank you. So thank you very much, Elizabeth. It's quite interesting to understand the triallelic and the biallelic. Really, it's fascinating. Now, I just wondered now for our women, what should be the stats that we give them? Because when we are counseling, and we're counseling at a stage where they've had a high chance on their combined or quadruple test, and they're now thinking of going either for an NIPT or CVS or amnio. So at that stage, I find women sometimes wanting to decide whether they should wait until they can have amnio or if they want to have the CVS right there and then because they don't want to have to wait for the 16-week period when they start the amnio. What should be the stats that we're offering them? Like, how accurate should I inform the patient CVS is? Or should we just be general and just say in general that it may be confined in the placenta?
Yes, I think we don't have specific stats. What I would say, once you get to the karyotype result, that is, if you need to go to that gold standard, that is diagnostic, and I wouldn't have any concerns about a karyotype result. I mean, you can nothing ever in this world is 100%, is it? But a karyotype from a CVS, I would say, is as reliable as an amnio. The one thing that I would always counsel is if you've had a high chance NIPT, I would really strongly recommend, where possible, waiting to have an amnio, because if you think the NIPT is testing the placental cells as well, so if you have confined placental mosaicism, you're going to get a high chance NIPT, and then you're going to test the same material and probably get the same result and have to wait for a karyotype. So in the long term, it's probably better just to wait for the amnio if you've had a high chance NIPT. Does that make sense?
Yes, thank you.
So I wish I could give, I'm a numbers person. I love to have numbers. I wish I could give you a number, but
Right.
speaking as, a patient as well as a scientist, I would say that it very much should be led on the family's personal experience of how quickly they want the result. I would say there is no need to wait for an amnio, as long as you accept you might have to wait for a karyotype off the CVS.
Right, thank you.
No problem.
Thank you. Gwenda, hello, Gwenda.
Hi, Karen. Just on that note where you mentioned obviously about if you get a high chance nipped and then suggesting waiting for an amnio. In terms of I'm just thinking of the standards as well. What's the national program's thought of that? Because obviously with our standards, I'm pretty sure, this is just off the top of my head, that if we get a high chance nipped, we need to offer and they have an appointment within like 3 days of, PND appointment within 3 days, I think. So of course that would suggest straight away generally that we should be referring them for CVS. So with you just saying that obviously you might want to suggest they wait for the amnio, I just wondered, has there been any talk about this with the teams in terms of?
Nadia, would you want to? I might be wrong. I might misunderstand, but I thought it was the target was to have the result discussed with the woman within that time frame, because obviously some women choose not to have any follow-up invasive testing following a high chance NIPT. But I think
yes, but I thought if they chose PND, it had to be done within a certain time, is what I thought.
Sorry,
I think Gwenda's right. So we have two metrics. One is after a higher chance NIPT result, they need to be seen within 3 working days to have the conversation and the discussion about options. But once they have decided to have PND, we say the procedure should be carried out within 3 working days as well. We don't set thresholds for that metric, but those are the time frames. I think in reality, and I think Beth, you did follow up your statement about waiting for an amnio. So it's really the conversation with the couple about, because some people waiting for an amnio is not going to be something that is acceptable. So we say, in terms of the guidance, that you offer PND, and that may be a CVS or an amniocentesis. I think what we're seeing playing out in reality is that by the time you get an NIPT result, particularly the ones that are having quads, you're getting it, the results are coming back on the cusp of whether they can have a CVS or an amnio. So the data is showing us that it's coming back around about between 14 and 15 weeks. So people are actually waiting and having an amnio in those scenarios. So I think we are seeing a mixture of things, but certainly if a woman wants a CVS, we don't say she has to wait for an amnio. She can certainly have a CVS at that point.
And it would still be diagnostic. I'm just saying that, and as we were showing in the majority of cases, it's not CPM. In I think the quote, in Nikki's results, which was actually a small sample size, so I think it's probably higher, 85% of them were confirmed. So you would have got that triallelic result from your CVS and it wouldn't have been a concern. It's just there's that very small number of false positives by NIPT, where if they were false positive NIPT due to CPM, you would get a biallelic trisomy and then you'd have to wait for a karyotype. So it's just something to be aware of. But the majority of times, by far the majority of times, you would get your diagnostic result from QF PCR from a CVS. It's just things to think about testing.
I would say that that's the important thing, is when you're having that conversation at that point with the woman, we need to be honest at that point and say, for a small proportion, I think it was 0.2% or something you said, Beth, the result will indicate that you have to wait for the karyotype. And I think that's the kind of honest conversation to have, rather than saying they can have only an amnio at that stage. Does that make sense?
Yeah, no, it does. It's just, it was more just the thought that, whether the parameters would change. But I think it's all just really down to the counselling, isn't it, really? And just making sure they're aware of that risk. And in the absence of any abnormalities on scan, it's the guidance that's already in written, isn't it? That we certainly
Correct.
suggest they wait for karyotype before TOP, for example. So
Yes.
Yeah.
And no two women are going to be the same. This is complex stuff.
Yeah. Thank you.
And yeah, I mean, and that's why we will accept either. I was just saying, you know, it's just something to bear in mind that if it is, if NIPT was due to CPM, you're going to get the same result in your CVS, but it's a small number of a small number, isn't it?
Thank you. We just have one more question. Yutundi, you got your hand up.
Yeah, just very quickly, I just wanted to confirm, like how Elizabeth said, that now you use the trophoblast in the core to actually get a better picture of the placenta. So is that just in Birmingham, or does that include St. George's and TDL?
It's in the best practice guidelines. So I don't want to comment on behalf of other labs, but I believe that they're all doing the digest rather than a single villus biopsy. And Nadia's nodding. I didn't want to say definitively on behalf of others, but I do understand that every lab in the UK offering the QF PCR will do this digest, so you get a fully representative picture of the placenta.
Thank you.
It's much reduced the number of false CPM results by doing that.
We've got a couple of other, we've got a couple of other hands. If people are happy to stay on the call, I know we're coming up to time. We'd just take another couple of questions. Is it Victoria? I can't see your name doesn't come up.
Oh, I put my head down. It's okay.
Okay. Let's see. I can't see on the way my screen's open.
Claire's the next hand.
Oh, lovely. Claire.
Hello. I have kind of one comment and one question. Something we've noticed is that as a lab processing samples, and so it's unusual, I think, as a screening coordinator, that we would look at it quite in this way, but something we have noticed is that women are, on some occasions, they're choosing not to have the R445 pathway and not to go for invasive testing straight off. They're choosing a CST instead because they want their PAP-A level, which is obviously an incidental finding, but because their trust that they're being screened at doesn't offer a separate preeclampsia screening pathway, which is obviously not part of the national programme, they're actually choosing to have a less good test for their trisomy screening so that they can have a combined, so that they can get their PAP-A. And I don't know really, I just feel like it's an interesting thing. I think it's something that kind of needs to be noted, and there's a comment just come up from Angela at Airedale saying the same. So that was kind of my comment, and my question is a bit of a random one. Oh, other people are now saying they have the same thing as well, so I felt like it wasn't very isolated. My question was, it's something we were discussing this week. We had a woman who had a pre-implantation genetic diagnosis of her own embryos, and she had a history of an embryo that had been tested and had been tested and come back as having trisomy 21. Obviously, then she didn't have that embryo implanted and have a pregnancy from that embryo. But our question was, in those instances, would that woman then be eligible for R445? Because strictly speaking, her egg has had a conception of a baby that has been diagnosed potentially with trisomy 21, even though she didn't then continue the pregnancy with that embryo. So it was we were basically like, I don't know. And then on further it kind of played out itself anyway, but so we didn't go to the lab to ask. But I thought this was quite a good opportunity to just kind of say, what do you do in that situation?
So it does say in the test directory guidance that if the trisomic pregnancy was through IVF and future pregnancies are from the same trying to think of a egg producer sound, you know, but from the same woman, then they are eligible.
Okay.
I would say, I mean, this is such a rare scenario. I would say, I think to the letter of the test directory, she would be. I feel a little uncomfortable because we all know that trisomies are more common in IVF anyway. So her risk probably isn't as high as a woman who's had a natural trisomic conceptus. So I don't think but I would say she would be eligible to the letter of the test directory.
Yeah, she's had a previous trisomy, yeah.
It was just, we were like, how far back do you go? So
Yes.
I thought that was an interesting case. That was my 2 questions.
Yeah, and lots of comments on PAPE, and of course it's difficult for us to comment on PAPE because it's a biochemical test, and we work in the genomics sort of arena, so a little bit difficult. I know there are different challenges with that out there, and of course, yeah, combined screening is a screening test for Downs, Edwards and Patels. It's not a screening test for fetal growth restriction, so I think we're all aware of that. Yeah, no pathway is perfect, is it? And it's about, yeah, we would advise that they're offered and given the choice. Let's have a look. Bonnie, you've got your hand up.
Hi there. I'm sorry we joined the call just a little bit late, but so I don't know if you wanted to save this for the end, but right back at the start, you said that 85% of the high chance NIPT results, 85% confirmed a high chance result on a QF PCR. What happened to the other 15%? Were they not confirmed, or sorry, we missed that little bit of information?
So it was I should say it was from a very small sample size. So it wasn't all of the 1,800 women who'd had R445. So there were just 3 women that we know of who had a high chance NIPT that QF PCR on an amnio didn't show the trisomy.
Okay, it did not confirm it. Okay, so it's like a false positive NIPT.
Yeah, but what I just want to flag that 85%, I think, in this patient cohort is
very small.
Within a bigger population, your false positives would be much lower. It's just because it was such a small sample size that 15% represents 3 women.
Okay, and so the next natural question to that is, do we have any statistics on the 1,800 women that had the R445, if there were any unexpected babies born with Edwards-Patels Down or were found later in the pregnancy to have those conditions, and through spontaneous fetal demise, you know?
We don't have that information here as a genetics lab. I don't know, Nadia, if that's something that's being collected like it is with the NIPT through this fetal anomaly screening pathway.
Not for the R445. So we're only following up the outcomes for the FAST NIPT.
At George's, we do follow up the low chance outcomes as well, but obviously it's not a very large number, and most women, because it's only been a year, will still be pregnant, so those outcomes won't be available. But we haven't had any who have had a low chance result that have been sort of postnatally diagnosed, but again, that's a really small number of women, but no false negatives, as it were.
That's lovely, thank you. And are we still awaiting the outcome of the evaluative rollout of the NIPT in general, or is that data been published?
Yeah, shall I just give a quick update on that?
That'd be great, Nadia. Thank you. Yeah.
So we've got 3 years worth of surveillance data, and when I say surveillance, I mean things around the test. And we've got 2 years worth of data on the outcomes of the pregnancy, which we took to the UK NSC back in June. The minutes of the UK NSC meetings are now published, so they're on the website. And like most things with the UK NSC, it's quite bland. It doesn't say very much, but what I can say is that it went through quite smoothly. But we are just finally waiting on the outcomes of the last year, pregnancy outcomes from the last year, which we should have by December. So we will know for certain what's happening by January. But we're just waiting on the final pregnancy outcomes for the 3rd year of the evaluation. But everything looks okay. We might make a couple of tweaks, but generally speaking, because of your hard work, really, it's gone very, very well and delivered really well. So that's probably as much as I can say at this point.
Thank you. That's really helpful. And I think what we'll do now is just end the questions here because we have gone over. So thank you for sticking with us. I'm going to just signpost you to some other resources. So I'll just share my screen again. And let's have a look. Okay, so hopefully you can see that, and there are lots of resources on genomics out there. So Genomics Education Programme website is a really good place to start. There's little bite-sized learning pieces on there where you start off what is a gene, what is a chromosome, and gives you information about study going right up to master's level. And there is still funding for anyone who wants to progress their genomics learning, whether it's one module or a postgrad certificate or a full master's. And certainly that's what I went off and did when I was a screening midwife before this role. So lots of information there, go and have a look. Obviously we've got the modules, the antenatal and newborn screening modules have got the NIPT modules there. Genomics 101 gives you a good grounding in genomics, and there are also modules in close relative marriage and understanding the genetic risk associated with consanguinity. FutureLearn have a lot of genomics modules on there, including one on genetic inheritance in pregnancy. Sometimes when you go on FutureLearn, it looks like there's a paywall, but actually that module in particular is free. And if you're working in fetal medicine or screening, I would strongly recommend it. I've done it, it's really good. G-Notes is expanding on a weekly basis almost, and there's a whole section on fetal and women's health, and this just gives you information that's concise, that's accurate, and that you can use in clinical scenarios. So for example, there are G-Notes on QF PCR, on the R445 pathway, on NIPT, on different conditions. So again, go and have a look, and that can really help you in clinical practice. There are a lot of resources out there on genomics, and it can be sometimes really difficult to know where to start. So we've developed the Genomics Learning Passport for midwives. And this is a Word document that you can download from the Genomics Education Programme website. And it just takes you through all the key learning resources that we think you should know as a midwife. Firstly, for all midwives, and then a second section for those of you in specialist practice. And you can edit it, you can add in your own courses, you can record your personal genomics learning journey in a format that supports revalidation. So it can just help you, guide you as to what you perhaps should do, what you should do next. So that's a useful document. We've also currently got a National Genomics Lunch and Learn webinar series taking place. So that's the QR code there for the previous webinar, so you can go and catch up on those. But we've also got the next two webinars coming up are one on genomic red flags in pregnancy, and that's in September, and then we're just giving an overview of the generation study, which is looking at genomics in newborn screening, and that's in October. So again, we're going to send these links out to anyone who registered for this webinar, but do sign up, and if you're interested, and share the links with your colleagues, because some of these are relevant for sort of all midwives, not just specialist midwives. So in the Lunch and Learn series, we're kind of focusing on the basics, really. We've done the questions, I think. I know there's a couple of hands up. There's a feedback form. It's really important that we get your feedback on these webinars. Find out what worked, what didn't work, and what you'd like in the future. And if you want more depth in any particular topic, whether it's a test like QF PCR or whether it's a condition, it doesn't matter, just pop it in there. Even if you think, oh, it's quite basic, but I want to know about this, then we can probably put on a webinar for you. So do let us know. It won't take long to complete the form. So if you've got a smartphone, just scan the QR code. That would be really helpful for us. Thank you. We've got a couple of hands up. I don't know. I can't now see who's on the call. Beth, if you've got time to answer a couple of questions.
Yeah, of course. Yeah.
Well, we'll carry on then if we've got questions, because I think it's an important forum, and we don't always get the opportunity. So let me just see who's first. Bonnie, you've got your hand up again.
No, sorry, I think it just didn't go down from last time.
It's a hand. No, thank you. Okay, no worries. Cassandra.
Hi there. It was just to ask the three people that the NIPT result wasn't right. They were all trisomy 13s. Is that something we should be sharing with women, or not, or are we to wait for the findings from the national rollout?
I would say wait. historically, I mean, I've been working on NIPT for a really long time now. The test performance for trisomy 13 and 18 did used to be slightly lower than it was for trisomy 21. So that was just a technical reason. They are now all very, very similar, being with about 99.9% specificity. But what we do know is that CPM occurs with a higher frequency for trisomy 13 and 18 than it does for 21. So it doesn't surprise me that there were no false positives for trisomy 21, but the false positives that there were for trisomy 13. But like I said previously, I think Nikki's data, because she was only able to get follow-up information from a very, very small sample size, that false positive is overrepresented in that cohort. It isn't normally 15% of trisomy 13s turn out to be false positives at all. So yes, there is a slightly higher chance of a false positive for trisomy 13, 18, 21 due to CPM, but not enough that I think I would want you to counsel your ladies differently.
Okay, thank you.
I would just say on top of that as well, from sort of a midwifery perspective, a high chance NIPT result for T18 or 13, it's looking at the whole clinical picture. So if you've got that and you know about the confined placental mosaicism and there's no anomalies seen on scan, definitely would be a conversation that you would probably explore in more detail with the woman. But if there are sort of associated anomalies, that's a different conversation. So it's just looking at the whole picture together, I would say.
Thank you. Gwenda.
I was just going to mention on this chat actually, so we've recently, just in the last few weeks, had a case where a lady had a high chance combined result for T21, went on to have the NIPT. The NIPT came back with a high chance of Edwards. So straight away we were on the phone to the lab and just kind of confirming, okay, what's the likelihood of this? And quite quickly, it may have been you, Marianne, actually, I'm not sure, who, they explained that, yeah, it looks like it could be CPM. And so it was quite useful because we referred for fetal medicine, but more for the fetal medicine scan initially. But I kind of warned her to say that it would probably be advisable to maybe wait for an amnio and not go ahead straight away with the CBS. And it was quite comforting to know that obviously when she went up to King's, they gave this exactly the same advice as us. They did the scan and then said come back in 2 weeks for your amnio. And of course the amnio came back normal. So yeah, it's just quite interesting.
It is, and we've seen that as well, where the NIPT shows a high chance for a chromosome we weren't expecting. But I would also just edge of caution, we have seen that where it has borne out that the biochemical screening gave a high chance for one trisomy where the fetus was actually affected with a different trisomy. So it does happen both ways.
Yeah, and I think this experience for us has made me also not advise and counsel differently as such, but because obviously with the NIP they have that choice to just have the screening for the certain conditions in the same way that we offer combined screening. So i.e. if it's a high chance for the combined result for T21 on combined, they could just have the NIPT for T21 and not T18 and T13. So it's quite interesting now that I can actually use this for information to say, in most cases the results are going to be reflective and hopefully be correct or whatever, but we have now had a situation where someone did have a high chance for a different one than expected, and that might kind of aid people's decision making in terms of what to screen for when they come for the NIPT.
And it looks like Nadia was about to comment on.
Thank you. Absolutely right. So we have actually updated the wording in screening tests with you and your baby. There's a new paragraph in there that talks about that possibility of getting a higher chance result from combined or quad, but the baby has another condition. And the session we had in February, we talked about the 5% of women who were choosing to only have NIPT for T21, because we have seen that in the evaluation. Now, this may be one of the things that we definitely change, but it's a good point to talk about it because we know that the markers for combined are indiscriminatory. So most of the T13s we will actually pick up via a higher chance T21 result. So please, when you're counselling the women for the NIPT stage of it, it's really advisable if they're going to have NIPT that they have NIPT for all 3 conditions, rather than perhaps saying, oh, but you're going to have a scan anyway and we're going to pick up the T18s and 13s on scan. It would be better that they have NIPT for all 3 conditions. And that's really important because we've all learned this lesson throughout the evaluation.
And just to say that, you know, if you had a high chance on your combined screen and went on to have an invasive, you would have all 3 for exactly that reason. So it's, you know, it's a logical follow-up.
Thank you. I think we'd just take one more question, if that's okay. Bonnie, I'm assuming that's a legacy hand.
Yeah, I think it's Susie. It's got her.
Susie, yeah.
Yeah,
Susie,
still
hi.
legacy, sorry.
Hi, Susie,
I just wanted, yeah, sorry, I just wanted to pick up just on what Marianne was saying about scan findings in association with your NIPT. That I think one of the limitations with that is with R445. I mean, we actually just don't have the capacity here to offer the earlier scan anyway. But one of the things that women miss out on if they have the earlier scan, it is much harder to pick up on a raised nuchal or any anomalies when you scan at sort of 10 to 11 weeks, even 11 to 12 weeks, really. And so you're not really able to make that consideration alongside the NIPT request when you do that test earlier, at the earlier gestation.
I think that as well is reflected in some of Nikki's data when she was showing, because although we can accept the referrals from 10 weeks, the majority of women weren't coming to us till 12 weeks or so because people were fitting it into their screening pathway. So just because we can accept for 10 weeks, I think a lot of centres wait to have that routine early scan anyway and take the NIPT blood instead of the combined screening blood, if you see what I mean. They're just doing it at the same time and they would have done a biochemical screen. So just because you can do it earlier doesn't mean you have to do it earlier.
Yeah, we understand ultrasound capacity is tight in many trusts, so you have to work with within your local constraints, I guess.
But we do have to be quite hard line on that, having to have had a scan, because we do know that one of the reasons for false negatives is a low fetal fraction. So we need to make sure that those women really are only being tested once their gestation has been confirmed.
Thank you. We'll call it a day there. Thank you everyone who attended. Please, can I just highlight the feedback form, if you could do that. I can't tell you how helpful that is to us. And if this has been useful and you want more sessions, this is the way to sort of tell us what you'd like, or you can email us separately. Like I say, the recording will be available and we'll email out to everyone who registered, and we'll try and make the slides. I just need to check with my fellow presenters. Just to check that we can give you access to those as well. But again, we'll let you know. So thank you, everyone. Yeah, don't forget the feedback form. Thank you.
Thank you.